Hi everyone, this is Ms. Ramani, and for today's lesson we're going to learn about genetic mutations. The concept of genetic mutations is actually one that has been popularized quite a bit in comic books and in mainstream media. However, the way the mutations are represented is not usually realistic. The idea that changes in our DNA can give us superpowers is appealing, but, and I'm sorry if that disappoints some of you, it is entirely unrealistic. And so mutations are basically changes to the DNA code. They are not the only type of genetic abnormalities or not the only cause of genetic abnormalities. Changes in the number or in the structure of our chromosomes, something that we call chromosomal abnormalities, the topic of what happens when you have extra chromosomes, too few chromosomes, or pieces missing of chromosomes, and so on, that was covered in the grade 11 course. Today's lesson will focus only on genetic mutations. So let's begin by taking a look at the locations of mutations and where they can happen. So first, let's take a look at where they can happen within the body. And there are two main places where mutations can happen within the body. The first type are in the somatic cells, and so we call those somatic mutations. Somatic mutations are mutations that happen in our body cells. So you might think, well, isn't all our cells our body cells? Well, as far as biology is concerned, we differentiate body cells, which are somatic cells, which are basically most of the cells in your body, from the cells that are found within your sex organs, the cells that are found in your egg-producing ovaries or your sperm-producing testes. So somatic mutations are basically the ones that happen in any of your cells in your body that are just going to undergo mitosis and are not the result of meiosis. If you have a somatic mutation, it'll have an effect on a part of your body that is affected by the mutation. The cells that have the genetic mutation are going to be the progeny of those cells with the mutation. Somatic mutations can happen in the womb, but most often they happen once you are already born and throughout your lives. And this is something that we contend with constantly. And oftentimes these types of mutations just lead to cancers, really, if anything at all. The other type of mutation, which is the one that is most concerning in terms of lifelong effects and more severe effects, are what we call germline mutations. And these are the mutations that occur in our sex organs, our egg-producing ovaries or our sperm-producing testes, or if they occur on the embryo really early on in development, so maybe in the zygote or one of the earlier stages of embryonic development. And with germline mutation, the entire organism is affected by the genetic mutation, because if the mutation was on the sperm or the egg, then every single piece of DNA that the organism has will have that genetic mutation. And so germline mutations are more dangerous because they don't just affect a little tiny part of your body, it will affect your entire body. And oftentimes these are the types of mutations that lead to genetic diseases or genetic disorders. Germline mutations are also the reason why whenever you go get an x-ray taken, they will make sure that they'll cover your genital area with a lead bib or a lead blanket, because any mutations that can occur in your eggs or sperm cells or your egg and sperm producing organs could have possibly lifelong and serious effect on your offspring in the future. So we've categorized the different effects that genetic mutations can have depending on which types of cells they occur in. But how about we take a look at what are the different effects of genetic mutations depending on where they occur within the DNA molecule itself. And so that is the difference between mutations that occur in sections of our DNA that we call coding regions versus sections of our DNA which we call non-coding region. And it might surprise a lot of you to learn that, as a matter of fact, most of our DNA is what we consider to be non-coding DNA. Some estimates estimate that about 99% of our DNA is actually non-coding, with only 1% or so of our DNA coding for genes and coding for proteins. When scientists first figured that out, when they finished the Human Genome Project, they were extremely surprised to find that so much of our DNA was actually what they at the time called junk DNA, because they didn't know what it did. They just knew that it did not actually code for proteins, and so therefore vast amounts of our DNA was not containing the instructions for our bodies. So imagine if we go back to that recipe book analogy that I gave you before, and 1% of the recipe book was actually recipes, and 99% of it was what at first appeared to be just nonsense that did not contain any recipes. And so when DNA mutations happen in what we call non-coding regions of the cell, the effects of those genetic mutations are going to be vastly different than when they occur in the genes themselves. So they can happen in the vast areas of the DNA molecule that are in between the genes. But even within the genes, if you remember from our last lesson, we have both exons and introns. So sometimes you can have mutations that happen within introns, within the gene itself. And so those types of mutations are not going to be as detrimental to the organisms as mutations that happen within the actual genetic code that codes for proteins, within the exons of genes that are then going to be interpreted and read and transcribed and translated into an actual protein. And when we take a look at the junk DNA, though, just to clarify again, so I don't want you to kind of come out of this lesson with an idea that most of our DNA has no purpose whatsoever. There's many different purposes that we've so far found for a lot of our DNA. And this is an area of biology and genetics that is yet to be fully explored. There's a lot that we still need to learn about that. But one of the things that we've learned is that, first of all, some of that non-coding DNA can be the introns within the coding sequence. So those genes that have those introns within them that are not necessarily going to then be part of the mRNA transcript that exits the nucleus for translation. But then there's other sections. For example, one thing that we find is that a lot of our DNA is composed of these things called repetitive elements. And repetitive elements could be, say, for example, the genetic code GAC, repeated maybe hundreds and hundreds or thousands of times. And we don't know why that is, but this is an area of DNA that is very vast within our genome that just happens to be there for so far unknown reasons. We also know that some of our DNA that is non-coding is actually the remnants of viral infections from the past. So these are types of viruses called retroviruses. These are viruses that can, when they infect our cells, they insert their DNA code within our genome itself. And anytime that that has happened within the history of humanity, and not just humans, but our ancestors within the history of our evolution, if those retroviruses inserted their DNA within germline cells, then their DNA ends up being part of our genome today. So a lot of our DNA is also DNA that came from viruses that infected our ancestors, and going all the way back to the earliest cell. We can actually do DNA analysis in order to determine the level of relatedness between species. And one of the things that we look for sometimes is those viral pieces of DNA, because if you find similarities in those retrovirus, then you can determine whether or not they are related to each other to a certain degree. Some of our DNA that is considered to be junk DNA actually has a very important function as regulatory sequences. So these are actual pieces of DNA that may not code for proteins, but they might code for, say, tRNA that are involved in translation, or microRNA that are involved in regulating gene expression, or even some of the proteins that are involved as transcription factors or something like that. So these are sequences of DNA that may not code for genes that produce proteins that we use in our body, but they might code for other proteins or RNA that is used during the regulation of our genetic expression. And then last, we have a lot of what are called pseudogenes. These are genes that are not turned on anymore. These are probably the remnants of our evolutionary history, genes for traits that we no longer carry, that we no longer express, but their history is there within our code. So there's a lot of reasons why we have all this junk DNA. And last, also, we have areas of our chromosomes that are filled with repeated sequences that are non-coding, that have actually a purpose structurally within the chromosome. So for example, the centromere, where the two chromatids attach in the center, that's a lot of non-coding regions of DNA. And then at the ends of our DNA, we have vast sections that are called telomeres. Telomeres are vast sections of non-coding sequences that serve a purpose to prevent the destruction of our genetic code as we age and as we reproduce. So when mutations happen on 99% of our DNA, which is the non-coding regions, they are going to have very little to zero effect on our cells and our bodies because they're not the parts of the DNA that actually contain our genetic code that codes for our proteins. So let's focus then on what happens when mutations happen on that 1% of our DNA that is coding. We can actually categorize the mutations into a couple of different types. The first type is what we call point mutations. And point mutations are basically mutations that occur on a single base pair. Because the DNA molecule is double-stranded, whenever we take a look at mutations, we have to take a look at what happens to not just a single letter, but also its complementary base. So it's always going to be done in pairs. And there are different types of point mutations. First one is something called a base substitution. Substitutions, as the name implies, are basically when one letter is replaced or substituted with another. And so there's two types of base substitutions. The first one are called transversions. Transversions happen when a purine is replaced with a pyrimidine, or a pyrimidine is replaced with a purine. So for example, in this particular example, you have an initial sequence with a thymine, and the thymine has been replaced with an adenine. That pyrimidine, which is thymine, is now an adenine, which is a purine. And another type of mutation, though, is what we call a transition. A transition is when a purine is replaced with another purine, or a pyrimidine is replaced with another pyrimidine. So for example, you have a guanine that is being replaced by an adenine. Guanine and adenine are both purines, so you're replacing a purine with another purine. Another type of point mutation are what we called insertions. Insertions, as the name implies, is when we are inserting something within the code. So this is not substituting or replacing a base with another base. This is actually introducing another letter within the code itself. So where the arrow is pointing right there, what would happen then if we were to add a nucleotide pair right there in the middle of that code? And so that's an insertion. It basically makes the DNA strand a little longer by one base pair, and it can have severe effects on the genetic code itself. The opposite of an insertion is what we call the deletion. And so let's say we remove a pair. So for example, let's remove that cytosine to guanine base pair. And so now the DNA strand is a little bit shorter by one base pair. And that is again going to have some effects on the genetic code and the proteins that it codes for. Non-point mutations can also occur. These are mutations where you have more than one base pair affected. So you can have, for example, the substitution of more than one base pair, or the insertion or deletion of two or three or more base pairs. And so the consequences of any of these mutations, the base pair substitutions, insertions, or deletions, can vary depending on the type of mutation that occurs and what effect it can have on the actual genetic code. So let's take a look at that next. What are the effects of those mutations on the protein sequence itself? So for the next few slides, we're going to take a look at several different effects that changes in the genetic code can have on the protein sequence. And each of the next few slides are going to look very similar to this one. On the left-hand side, you have in purple the initial non-mutated sequence of a strand of DNA. Notice that it's double-stranded, right? Because DNA is double-stranded. But then you see beneath it the mRNA, the single-stranded mRNA that DNA sequence codes for. So transcription has happened, and we have that section of mRNA, and we're going to then show what that altered sequence would look like on the mRNA, which in turn is then going to be translated into a polypeptide, an amino acid sequence there in blue. I've also added the genetic code so that we can kind of follow along to see what the effects of the genetic mutations are. So let's start with this first genetic mutation. So you can see here, the mutation has replaced a thymine adenine base pair with a cytosine guanine base pair. So this is a base substitution. And in the initial sequence, the original non-mutated sequence, that particular genetic code would have been transcribed into an mRNA that had a codon UCA. Remember that the genetic code is read in 3 letters, and each of the 3 letters is called a codon. And so when those 3 letters are then translated into an amino acid sequence, take a look at the genetic code table at the bottom, then UCA would actually code for the amino acid serine. So serine, SER, that's its short form, it's found right here. Now, in the mutated version of the codon, though, we no longer have UCA, we now have CCA. And CCA no longer codes for serine, it actually codes for proline. So now we have one altered amino acid. So in this type of mutation, which we call the missense mutation, a single amino acid has been changed due to usually a single base pair substitution. We substitute one letter in the initial sequence of the genetic code, which in turn leads to a different codon that is going to code for a different amino acid. So we call these missense mutations. Another thing that can happen when you have a base pair substitution, so again, here we go, we're going to substitute a different base pair. In this particular case, the thymine to adenine is going to be substituted into a cytosine to guanine, but it's a different one. And in this particular case, then, the codon that is affected is the previous codon, which is GGU. Now, GGU codes for the amino acid glycine. That's the original code. Now let's take a look at what happens after the mutation. So in this particular case, after the mutation, the codon GGU is now GGC. Let's take a look at it, what effect that has on the amino acid that is coded for. Well, GGC, well, it also codes for glycine. So this particular type of mutation actually has no effect on the amino acid sequence, and we call that a silent mutation. Same type of mutation that occurred in the previous example with a base pair substitution, but because in the previous example it was the first letter in the codon that was substituted, that actually led to a different amino acid being coded for. But because in this particular case it is the third letter in the codon that is substituted, and oftentimes the third letter in the codon does not really matter in terms of determining what amino acid is being coded for, because many amino acids are coded for by more than one codon, and it is usually that third letter that might differ between them. So in this particular case, glycine is coded for by the codon GGA, GGC, GGU, and GGG. So any 4 of those codons would still code for glycine. So let's take a look at another mutation. In this particular case, we have now another base substitution. We've substituted this cytosine to guanine base pair for this adenine to thymine. In the original codon, we have a codon UGC, which codes for the amino acid cysteine right here. Let's take a look at what happens after this mutation. The new codon is UGA. That's very similar to UGC, but in this case, changing that third letter in the codon actually does not lead to the exact same amino acid as the previous code, but it actually leads to a stop codon. And so with a premature stop codon, now the sequence is going to be terminated. When the mRNA is read by ribosomes, it's going to read a stop codon very early on in the mRNA sequence and is going to terminate translation. And a polypeptide that might have been maybe 1,000 amino acids long might now just be 10, 15, 800, but is going to be shorter. And so whenever that happens, we call that a nonsense mutation. A nonsense mutation is a mutation that causes one of those 3 stop codons to appear somewhere within the genetic code well before it is meant to appear, which shortens the length of the polypeptide. The opposite of that would be what we call the sense mutation. A sense mutation would be one in which a stop codon is lost. So this would be a mutation in which, say, the original code might have been UGA, which coded for a stop codon, but now let's say, for example, that changes to UGC, say, and then so now instead of having a stop codon, we add a cysteine amino acid, and then the mRNA is going to continue to be read and adding more amino acids. So you end up with a protein that is longer than the original protein. And so having a shorter protein or a longer protein sequence is going to be detrimental to the shape of the protein and therefore the function of the protein. So let's take a look at what happens when instead of a base substitution, we have something called an insertion. And so in this case, let's add a base right here in between that guanine to cytosine and guanine to cytosine bases. Let's add one base pair right there. And so the original code would code for the codon GGU in the original sequence, which codes for the amino acid glycine. And after the insertion, we now have an adenine in between those two guanines. So instead of being GGU, the codon will be read as GAG because now there's an extra letter in between those two guanines. And because ribosomes read the DNA code 3 letters at a time, regardless of what it was meant to be initially, it's not going to count the fact that adenine shouldn't have been there. It just reads the next 3 letters. So the next 3 letters in that mRNA sequence, instead of being GGU, is now GAG. And so GAG codes for glutamic acid, which is a completely different amino acid. But then let's take a look at what happens to the code after that point too. Because the next codon in the original sequence would have been UCA, which would code for the amino acid serine. And in this new code, because we added an adenine in the previous codon, then the uracil in that codon, instead of being the 3rd letter in that codon, it becomes the 1st letter on the next codon. And so instead of having UCA, we now have UUC for the next codon. And that codes for the amino acid phenylalanine. And then every single codon after that is similarly affected. So essentially, by adding a base pair in the middle of a genetic sequence, we've essentially shifted the reading frame of the mRNA so that the codons are read off by 1. And so we call this frameshift mutations. And frameshift mutations, they essentially cause every single amino acid from the point of the mutation on to be different than the original code because of a shifting on the reading frame of the mRNA sequence, because ribosomes read mRNA 3 letters at a time regardless of what the initial sequence was supposed to be. Frameshift mutations do not only happen when you have insertions, they will also happen when you have deletions. Same effect, adding a base or removing a base will essentially shift the reading frame of the mRNA so that codons are going to be off by 1, or if it's 2, by 2 from that point on. The only time that frameshift mutations are not as bad is when you have 3 bases that are inserted or deleted, because the codons are 3 bases long. By inserting 3 bases or deleting 3 bases, we're essentially just either deleting one amino acid or inserting an extra amino acid, which could have a very bad effect but may not be that dangerous to the organism as a frameshift mutation is, which essentially creates a completely different protein from the point of the mutation on as was the original intended. So there is no way that this new protein is going to work at all like it was intended, because it is essentially a completely different code from the point of the mutation on. So just very briefly, let's just summarize the different types of mutations and their effects. Substitutions can lead to missense mutations, silent mutations, nonsense or sense mutations, but they never lead to frameshift mutations. Whereas insertions and deletions will always lead to frameshift mutations of some form, unless it is 3 bases that are inserted or 3 bases that are deleted. But these frameshift mutations themselves can also lead to nonsense mutations if a stop codon appears too early as a result of the frameshift, or a sense mutation if a stop codon does not appear early enough as a result of the frameshift. So let's take a look now how mutations can affect the entire organism. There's a variety of different ways by which mutations can affect an organism, but in general we're going to either see maybe something like a morphological change, which is an obvious physical change in characteristics of the organism. Biochemical changes are not as obvious, they're inside the body, but for example, in the case of cystic fibrosis, a single base pair substitution leads to a missense mutation with a single amino acid change in the CFTR chloride channel, which now produces a mutant and non-working chloride channel protein, which is the cause of cystic fibrosis. And of course, mutations can sometimes be lethal if they're completely incompatible with life. The causes of mutations are basically twofold. They can either occur spontaneously during errors that occur naturally during DNA replication that are either missed by the last step, which is the proofreading step of DNA replication, and therefore accumulate in the cell. And this is one of the main causes of both aging and cancer, is just natural spontaneous errors in DNA replication. But they can also be induced by our environment. So these are what we call induced mutations. And induced mutations can come from a variety of different forms or environmental agents called mutagens. And mutagens can be things like radiation, like UV radiation or X-rays, chemicals in our environment, chemicals in our food, or even infectious agents like viruses or bacteria that can actually cause genetic mutations as well. So there's a variety of different things that can cause mutations. And so that's the lesson for today. I will talk to you later. Bye.